their other time. But Team Alpha, agreed to take this earlier time instead of their original time.
So yeah, while we wait for them and one more judge to come back, I'll just like to say that the scoring is going to be mixed between what the team submitted in their teacher's manual as well as their pitch performance today. So Izzy, Lisa, Ruby, and Alice are contributing 50% of the marks for the live pitches, and the other 50% comes from our team that's busy judging their teacher's manual. And the teacher's manuals are really important because we can take those and immediately start using them in the Philippines. We might have to make a few edits, but a lot of the teams did a very good job on those.
So I've just led Team Alpha, so Team 9, into the meeting. So like I said, thank you so much, Team Alpha, for switching last minute with the group that couldn't make it. I think you only have 3 members who are in. Are you waiting for anybody else, or is it just you 3 presenting?
It's just us 3 on the Zoom chat.
Okay, in that case, you should have permission to screen share, so you can start whenever you're ready.
Hello? Hi, can you hear me?
Yep,
we can hear you.
Hi. Very good afternoon to our judges, Ms. Herrera, Ms. Calda, Ms. Sola, Ms. Kwal, and our mentor, Mr. Moy. We are last but definitely not the least. Team Alpha, we present to you our project, The Eye of the World. The Eye of the World is a DIY makeshift microscope. I'll be speaking a little bit about how our project correlates with the needs of children in the Philippines, as well as the United Nations Sustainable Development Goals. Looking at the Earth and Life Sciences curriculum for high school students in the Philippines, it requires the use of microscope or hand lenses with at least 5 times magnification. So it came to our minds that high school students in low resource communities may face issues with access to expensive scientific instruments like a microscope. Therefore, we came up with the idea of a DIY makeshift microscope where we use a spherical water droplet as our magnifying lens. Looking as simple as it is, the physical theories behind are related to optics, which fits well into their physics curriculum. The design of our project complies with the United Nations Sustainable Development Goals number 4 and 12. We hope that quality education can be provided for all, and to achieve that, we hope scientific instruments are accessible to all. The Eye of the World is highly affordable and sustainable. We didn't even have to fill in the public sciences reimbursement form because the Eye of the World is made up of solely household items that would have gone to trash. The materials we used are as follows. They're affordable, accessible, and reusable. The preparation has to be done by teachers are very minimal. Teachers only have to gather the materials and students can work with groups of 4 and follow simple steps, which now I'll pass over to Takuma to demonstrate.
Thank you, I'll take it over from here. So as you can see on the screen, the materials are very simple and the structure itself is very simple. You can see the details in the manual itself, but I'll show you the actual item, our project, the final project later. Would you mind going to the next slide? Jane. So here are the instructions. Of course, the details are in the teacher's manual, but we're assuming that it's going to take about 1 hour for a group of 4 students to create a 1 microscope. And yeah, next slide, please. So this is our actual footage of a microscope in use. So it was difficult to calculate the magnification through mass, but we measured the actual sample and measured the image that we received. It turns out that this microscope can do about times 100 times magnification. And it's much easier to see on a screen, but it is projected on the wax sheet so that you can view, multiple people can view at once. Next. So the science behind it is pretty high school level physics optics. And this microscope differs from the Leeuwen Hooke's microscope, which is known as a very first microscope invented. And also it differs from a very common compound microscope. And as this mechanism is also explained in the teacher's manuals too. So finally, we're hoping to inspire the students to investigate their field of synthetic chemistry. They can analyze the shapes of crystals. They can test it on their hairs, their cheek cells. They can test their plant cells and animal cells. And even microorganisms, we figure that this is strong enough to look at the multi-cell microorganisms and some of the unicell microorganisms. And we're hoping that this project will inspire students to build more scientific instrument from the available resource and further expand their scientific community around them. And now I would like to demonstrate the actual microscope. So this largest part is the lens part, and you put the water inside this lens, and this clear tape here is a sample that goes over the lens. This is a pinhole, and then this goes into the lens, and then finally this is the screen that is used to project the items on the image on the screen. So if you could look at the previous slides, we've shown you how it looks like, and hopefully if I got enough time, I can show it here right now.
You don't, unfortunately. But the judges might be able to ask you to do so in their questions. So thank you very much for the presentation. Judges, who would like to start? Ruby.
Oh yeah, like if you could project it, because I was gonna ask if you took some pictures, because you said you were projecting it. So one way to actually demonstrate it to us, at least for this time being, if you have like pictures of what you actually took from your microscope, or if you have time now, was that Marcus that was talking earlier? Or Takuma, yeah.
Yep.
Is it quick to show, like the sample you have already in your glass slide?
Yeah, I can try it. So this is the microscope, and here's some water. You just use your finger and tap the water onto the lens, and then you put the sample over it. And we changed the design so that the sample just have to go over the lens so that it can project without adjusting the focus. And hopefully I can show it on the camera. If you had a table and you can place the light source on, it will look much more stable. But
okay,
yeah, this is what it looks like.
Hi there, it's Izzy here. I may have missed it, but I'm just wondering specifically, how did you adapt the design of the microscope for a low resource setting? Like, what were the elements that you changed?
Does anybody want to answer that question?
I can, oh, yeah, Takuma, go ahead.
You're on mute.
you were muted.
So we initially started from the Leeuwen Hooke's design, where they have a single lens, and it is practically made in 16th century, and it's quite simple design, but then it's really hard to view because of the limitations that it has. So Leeuwen Hooke's design, you're viewing the virtual image, and that requires you to be really close to the lens itself, while the compound microscope adapts this and creates a real image first and then the virtual image, which makes it much easier to view.
Okay.
But then having a second lens is very costly. So we tried to come up with a ways of having a water lens, but also have the projection of what the real image was. So we developed the screen, but then having the screen itself will not be sufficient in adjusting the real image being projected. So we then added the pinhole too, which is essentially a hole in the paper.
So these are all your adaptations. These are
Yep.
all things that you did without sort of. Okay, that's interesting. I was just wondering which part of this design was your contribution to the design.
Yeah, what again was the material used for your lens? And did you try other materials?
Yeah, so the lens itself is a water droplet that's on the hull. And we did try oil and the glass beads where it's not attainable, but in theory, it will work just fine or even better than the water droplet. And I personally tested it with the sugar water, and it does increase the magnification because of the higher reflection index.
Okay, thank you.
We have time for a short question, if there's any. All right, seeing none, thank you very much. And thank you again for switching your schedule for us. You're definitely not the last or the least. So we're going to prepare for the next group now. So I'll see you all later at the closing ceremony. So thank you again for your hard work this weekend.
Thank you.
Thank you.
And judges can use this time to enter their scores, please.
I am now letting in Team 7, so also known as MCFAM. So you guys should have permission to share screen if you want to start doing that. Otherwise, Leo will give you the thumbs up when you can start presenting.
Sounds good, thank you.
All right, thank you, Group 7. You may begin.
Okay, hi everyone. My name's Christine and I'm here with my friends Mary, Eliza, and Mariah. And today we are going to present our experiment on particulate behavior and textile effectiveness. So we wanted to look at a couple problems in the Philippines, and these prominent issues include air quality and textile waste. This is just a little graphic here showing how poor the air quality is in the Philippines, and we wanted to make sure that our experiment works to combat this. So specifically, our experiment assesses different textile pore sizes in their effectiveness in filtering different states of matter. So our experiment actually has 4 different components. So the first component is a mechanical lung model, and that will focus on targeting the curriculum of General Physics 1, as well as the curriculum of Earth and Life Sciences, specifically Area and Force for physics, and then organ systems for life and Earth Sciences. The next part of our exercise is a distance particle exercise, and that will focus again on the General Chemistry 1 and 2 curriculum, specifically looking at arrangement, spacing, and motion of the 3 different states of matter in space. And then the next part of our exercise is a data collection exercise, and that focuses on the General Physics 1 curriculum, looking at random and systematic errors. And the last part of our exercise is a textile exercise, and that will target the Chemistry 1 and 2 density of liquids and solids. So looking at our materials, these are the materials here needed for our different experiments and exercises. So Vaseline, water, solid and gas, balloon, plastic water bottles, different types of textiles, as well as different sheets of cardboard and paper, and then a spray bottle and a squeeze bottle. So I'm going to have my friend Mary here give a live demonstration of the mechanical lungs, the particle exercise, and the data collection exercise right now. I'm just going to stop sharing my screen so she can give you guys a demo.
Can everyone hear me?
Yep.
So Mary here with the live demos. First up is our little mechanical lung here that you can see. It's just a plastic bottle with a few balloons attached to it, but we'll be using it to show students the anatomy of the respiratory system. Pushing in to the bottle results in the balloon inside shrinking, pulling out results in it expanding, and that's basically how the lungs work. And this will be a precursor to our air particle spread exercise, which I will demonstrate now. So as you can probably see in front of me are 3 pieces of construction paper tucked into clear binder sleeves covered in Vaseline. All of these objects are easily found at most stores and quite cheap to buy. And these will actually act as our indicators. As for our source of particles, I will be using this squeeze bottle filled with flour. So I'll give it a squeeze and let's see what happens. Okay, hopefully some of you saw that. I don't know how it appears, but there was a nice little squeeze of flour into the air. Let's try it one more time to make sure everybody can see. There we go, that's a better cloud. So as you can see, the flour is kind of stuck to this indicator now because of the Vaseline. And students will be actually applying a randomized sampling approach to determine the percent cover of flour on these indicators using this as a visual aid to the percent cover. And they will take the average of 10 of the grid cells that are on these construction papers. It's a pretty common technique that you see, especially with tree canopy cover and cloud cover. And just before I go, I do want to show you a close-up of the indicators. There you go. All right.
Perfect. Thank you, Mary.
Anytime.
So just going back into the remainder part of our little pitch here, I'm going to wrap this up really quickly. So that was the first 3 exercises. So the last exercise we have is basically testing solid, liquid, and gases. Throwing through a textile when you spray it through the textile shown here and seeing if we can see it landing on another sheet of paper, and that can help inform porosity and textile size. So lastly, we wanted to make sure that our project looked at the Sustainable Development Goals, specifically goal number 3, good health and well-being, number 11, sustainable cities and communities, and goal number 4, quality education.
Thank you very much.
And that's all that we have today. Thank you.
All right. Ruby has the first question.
So yeah, I'm happy to see how you related that textile particulate into your experiment, but it wasn't, do you have any data on how the paper looks like when you have the different textile into the squeeze bottle? I kind of missed the connection there. So I guess your premise, yeah, go ahead.
Oh,
No,
so using the different textiles, we're trying to see the porosity of different liquids, solids, and gases to go through it. And that can be helped to inform how maybe different textiles could be used to make masks to protect against air pollution or potentially COVID.
Okay. So the way, I guess I'm looking for the quantification so that students can see the, in a way, final, so the way you quantify how much particulate got into the different, into the cardboard is by looking at that chart. Is that correct? The pre-calibrated, for the percent coverage.
Yes. I think Mary is about to talk about it right here.
I can actually go over that. So this is what, let me just show you how the demos, some of the demos look. I don't know if you can see that, but it is
Yeah,
covered in flour here. And there's a grid drawn on these construction papers. And what students would be doing is picking randomly cells from the grid and using the diagram here.
I see. Okay, it's clear now. I see.
How much of the cell is being covered. And we wanted to do that. We wanted it to be randomized because with the spray bottle, your trajectory of how you're pointing it can affect the results or how much the construction paper is being covered. So by randomly sampling, we can account for that possible bias.
There are grids. Okay. Yep. Thank you, Mary.
No problem.
I have a question. I'm just wondering if you could explain more specifically about how your lessons connect to the Sustainable Development Goals. I feel like you kind of rushed through that part, and I just want to know what themes you're actually focusing your series of lessons on.
Of course. So for Sustainable Development Goal number 3, specifically that goal looks at good health and well-being. And there is one specific target, it's target 9, 3.9 actually. And that aims at by 2030, substantially reducing the number of deaths and illnesses from hazardous chemicals, air, and soil pollution and contamination. So we hope that our experiment can aim to first off educate about these issues, and then maybe potentially get students to think about how they can solve these issues by potentially using different fabrics to make masks to help protect themselves as a barrier, or other ways. But we want our experiment to kind of be like an opening conversation to discuss these issues.
We still have time for one more question for the judges, Alice or Izzy. All right, thank you very much for your presentation and your demonstration, and of course for all your work these past 2 weekends.
Awesome, thank you so much.
Thank you.
Yeah.
We'll be bringing in the next group now, so judges, please enter your marks.
I have just let in the next team, which is team 15, otherwise known as SUS Educators. So to the new team, Leo will give you a thumbs up when you're ready to go. Otherwise, you can start sharing your screen now if you have slides.
Awesome, thank you.
Just a general reminder to turn off your screen sharing when you do a live demo so that we can see that video larger.
If I talk while another person's doing a live demo, can everyone pin it so that that one will show up?
Yeah, I think we can do that. Just make sure the screen sharing is off. That's
okay. Yeah,
key.
for sure.
Thank you. Just waiting for 2 more scores to come in, but I'll let you know when you're ready to start.
Am I good to go?
You're good to go now.
So my name is Stanley. I'm from the team SUS Educators, and today we're going to be talking about water analysis using turbidity sensor. So the problem at hand here today is 7 million people in the Philippines lack access to safe water, and they struggle to provide sanitation services to new communities, which is why we're focusing on the 6th United Nations Development Sustainable Goal. The solution is, you might think filtrating water with charcoal, sand, or gravel, and that's what you thought. And although these are really important, our focus is water quality. And why? Because not enough people talk about it and value its importance. So our prototype is a turbidity sensor. At the bottom is an LCD display to show results. It's connected to Arduino, which controls everything. And in the middle of the picture, I hope you can see there's a little device with a squiggly line on it. It's a light-dependent resistor, which detects light intensity. So with the high school students, our first 90-minute session, we'll be building this filter system with sand or gravel. And the reason is because we want to differentiate between filtered water and unfiltered water. In our second 90-minute session, we're going to use basic circuitry, cheap electronics to build the turbidity sensor. And it does seem really complicated, but trust me, we have a comprehensive guide provided in our teaching manual. And in our third 90-minute session, we want students to become analysts. become scientists and really design their own experiments around the turbidity sensor and test their hypotheses. Our suggested experiment is to test which filters are better. So which is why we wanted them to prep the sample in the first session, use sand or gravel and see which one works best. And onto our materials. So there's a lot of them. Here are the prices. We've quoted all of these sourced from an online electronics store from the Philippines, converted them to Canadian dollars. And trust me, they're actually less than $20, surprisingly. Our learning competencies. So our mission today is to equip youth in low resource communities with the tech and knowledge to create infrastructure for testing sustainable fresh water. And our project overlaps a lot with the grade 11 and 12 curriculum in the Philippines. And it includes concepts like optics, electronics and circuits, turbidity, and transmittance. So before we get into the live demo, I really want to highlight a key concept that turbidity is just a fancy word for seeing how much light passes through a sample. And it's not necessarily a foolproof indicator of water quality, but because you can say, hey, there's dirty water, the results are really bad, don't drink it. But sometimes there's water that's just really clear and the sensor cannot detect whether there's some chemicals that are bad for you. That being said, though, we can filter all the water that we want. But if we don't test the water quality of the filter, we don't know how effective that filter is, which is why we want to educate students and allow them to have the potential to go to their own communities, test water from their local regions and say, hey, I use this turbidity sensor. Our water is actually not so good, even though everyone's using it. And we have data to back it up. So onto our demo. Let's turn to Faye's camera. If everyone can pin her video, that would be great. So on the right, there is some dirty water she got from her backyard. She's going to put it into the sensor. She's going to shine a red laser. We're going to tell the students not to point it into each other's eye, obviously. There's a light dependent resistor on the end. And then here we can see the output. And as you can see, the light intensity is 500 units. At the moment, that means nothing to you, right? But afterwards, we're going to filter that water through this filtration system. And we're going to get the water on the left, which is a lot clearer. Put it back into the sensor. Let's see what, turn on the light, and let's see what value we get. And excitingly, we do get a higher value. And what that means is that more light passes through the sample because how the turbidity really works is light scatters through the particles in the water. And if there's a lot of particulates, it means there's dirty water. And so the emergent beam, there's going to be less light. And so we get a higher number when we filter the water. So thank you for listening. I hope you enjoyed it. And I really look forward to answering any questions that you have.
Thank you for the presentation and the demo. Judges.
So just wondering whether your turbidity sensor is part of the activity that students will be doing, or is that already prepared by the teacher and the experiment is just all about, testing the turbidity of the water?
So the students will actually be building their own turbidity sensor in probably groups of 3 or 4. And then it's 3 parts, right? First, building the filter. Second, building the actual sensor together. And the third one being making their own experiments, all student-led.
And I guess the concept of how you measure turbidity will be included in that activity where they will be building the sensor.
Mm-hmm. Yep. How the detector actually converts light into electron.
Mm-hmm. Mm-hmm. Okay. Thank you.
No problem.
So I really think this is a very empowering exercise. I think it's wonderful that you're using, science to empower the students and to ask questions and give them some practical things they can do to challenge their environment and the health of their environment. I'm just wondering if you have any extensions to the experiment that you contemplate. If assuming this goes to the schools and is used, do you have any extensions or future areas that you might be able to develop out as well?
Yeah, so we're thinking of, so this is the first step, the most simplest device, the most simplest sensor you can get, just using light. And we're thinking of incorporating, more advanced like science tech that we currently use, such as like the spectrophotometers. Maybe we can engineer something that is simple enough. It probably will be more expensive, but we can engineer something along those lines and detect for specific chemicals that we couldn't do in this one, right?
Be a bit more targeted. Yes.
Exactly, yeah, because there's only red light. We could use other lights, yeah.
So it's just particulate matter essentially in the water. You're not saying what it could be, what the chemical composition could be of that matter. Okay, thank you.
Ruby and Lisa?
Yeah, so I'm just curious because they're building their own devices, and you shared like sort of the cost-effective materials, but at some point they're going to need to use a computer, right?
Yeah.
So I'm just wondering about how you were thinking through, that component of the design of your lesson.
So what we were thinking of, there's two options, right? With the Arduino, all we could do is send the kits with pre-programmed Arduinos with the code. The source code is in the teaching manual, but if they do have one or two computers at the school, the teachers can pre-program them there, and even like call each group up and say, hey, this is what the source code looks like, which line correlates to what line, and yeah. So the students wouldn't actually be programming. It would just be a copy and paste kind of thing, but it's still insightful to learn what the code actually is.
I feel like there's an opportunity to add that as a follow-up lesson, right?
Yeah.
Because you learn also from deconstructing code just as much as creating code from scratch.
Yeah, for sure.
Okay, we still have time for one more question if anybody else has one. All right, thank you for showing us that demonstration and for that presentation. And of course, for all your hard work the past couple of weekends.
Yeah.
So we will see you all hopefully at the closing ceremony.
Okay, thank you so much.
Thank you.
Thank you.
You're running a little early, so we will hold off a little bit and let the judges slowly enter their marks. We have two more teams remaining, and then afterwards, if you're on the live stream, stay on the live stream. At about 4:05, we will switch over to our closing ceremony, and of course, that's also where we hand out the awards. But in the meantime, you can go to puebloscience.devpost.com, and that's where you can find the videos by each team. So if you missed some of the earlier ones, you can go back and watch their 2-minute video. And there's also a big People's Choice Award prize that the teams can win after today. So be sure to check out our Dev Post page. And again, why don't I just give our great sponsors one more shout out on this stream. So it is Manulife, MBNA, and TD Insurance. They've supported this for the past 2 years, and really without their big support, we wouldn't have even started the hackathon for science education. And so we really like to thank them. And this year, that support was joined by the BEST program at York University, and we'd like to thank them for joining us as well to provide an extra little motivation with extra prizes to our competitors. And I have recorded the score, so we'll just hold on a bit for Sarah and the Penguins of Madagascar, who are coming up next.
So I think a few members of their team are already here, so I'll just admit them for now. But we can wait a little longer for the last couple of people. So this is team 10, Sarah and the Penguins. We are running a little early, so I think you're missing 1 or 2 members. We can wait a little longer for them. But Leo will give you a thumbs up when you're good to go. Actually, I guess the judges are done. So as soon as your team members are here and you're ready to go, you can start, and you can also begin your screen share now if you'd like.
Okay. I'm just going to be presenting. I'm just going to wait for
Rest my team.
And yeah, just a note that before you start your live demo, to turn off your screen sharing so that the live demo video appears larger on our screens.
Okay, I'll just be talking. So.
Okay.
Okay, I'll just start. So, have you ever asked the questions, what is yeast? What can yeast do? How is it useful? This high school experiment is going to demonstrate many of yeast capabilities, such as ethanol fermentation and food spoilage. And not only are students going to learn all about yeast in a fun and entertaining game of Jeopardy, they're going to learn that microorganisms as small as yeast, not even visible to the human eye, can start up a whole business in brewery or even biofuel, an extremely sustainable and renewable energy source. In the first 30 minutes of class, the teacher will provide some background information on yeast, and students are going to set up their experiment. So how this is going to work is that each station will have 3 plastic bottles, 1 cup of water, sugar, and yeast in separate containers. In each bottle, they'll add equal amounts of water and yeast, but different amounts of sugar. In order for anaerobic respiration to take place, which is basically yeast making energy without the presence of oxygen, balloons will be placed over the bottles, which will collect carbon dioxide. And students will find that the bottle that had the most sugar had the most inflated balloon. So actually, I just did this experiment 1 hour ago. It takes about 30 minutes to 1 hour for it to the balloons to actually inflate. And as you can see, the balloon, which is this one, had more sugar, and this one had less sugar. And so the one that had more sugar obviously collected much more carbon dioxide. And so students will then actually measure the concentration, they will measure the circumference of the balloon using string, ribbon, or really anything they have at the 30-minute mark and the 60-minute marks, and they will record their findings. But that's not it. Using the circumference, they're actually going to have to find the volume of the balloon. And so many questions and tasks, just drawing the graph for amount of carbon dioxide produced over time, will be asked of the students. So you're probably wondering, well, why don't they go ahead and just find the concentration of carbon dioxide produced? But the issue with that is that concentration is a measure of both moles and volume. So as the moles of carbon dioxide increase as the reaction proceeds, then the volume also increases as well because the balloon is expanding. And so the concentration actually will remain the same for any amount of yeast, any amount of sugar at any time point. Now, you actually might be thinking, so students are just going to sit and wait for their balloons to inflate the whole class? The answer is, of course not. While waiting for ethanol fermentation to take place, students are going to learn so much about yeast, all through a fun and interactive, engaging game of Jeopardy, where they will get to choose one of 4 categories, such as basics of yeast, ethanol fermentation, food spoilage, or outside the box questions. And each category will have question banks, and different points and prizes will be awarded accordingly. And the last thing I have to say is yeast isn't only beneficial, it's also a fungus and can be responsible for spoiling your food. So to demonstrate this aspect of yeast, in the last 10 minutes of class, students will listen to the teacher explain how yeast can also spoil your food as it feeds on the sugars of fruit, for example. So students will compare the difference in mold growth on mango, which actually has high amounts of sugar, and watermelon, which has low amounts of sugar. And the next day, they'll come back and they'll observe the mold on the food, which actually will take overnight or maybe even 2 days. But in the end, students will continue their activities, and the next day, they will be asked further questions such as observation and changes in inflation of balloons. And actually, the balloons will deflate since carbon dioxide will be absorbed in the balloon. Also, I actually did another experiment last night, and these are the balloons right now. So as you can see, the balloons actually deflated. And so they'll be asked questions regarding topics like that will make them think outside of the box. Now, not only is this an educational and interactive experiment for the students using accessible, very easy, accessible resources, students will hopefully learn more about the potential yeast has, such as producing biofuel, which again is a very cost-effective energy source. That's everything. Thank you.
Thank you for that. Alice?
Yeah, I might have already forgotten about my chemistry, but can you remind me how fermentation would be producing CO2? And as I know, if you're using anaerobic digestion, you are generating methane?
So anaerobic respiration, so basically anaerobic respiration is a way—
Or respiration, not digestion, okay.
It's a way that yeast use basically can metabolize sugar without the presence of oxygen. And so they basically go through the process of glycolysis where they have glucose, which is, or fructose, sucrose, any type of sugar. And basically they're gonna like metabolize the sugar and they'll produce like a compound known as pyruvate, okay? And basically it's without oxygen, usually normally in the presence of oxygen, pyruvate goes through like different transport chains, the Krebs cycle, electron transport chain, all just to make energy, which is ATP, adenosine triphosphate. But when there's no oxygen, that actually can't happen. And so the pyruvate, which is what is produced, basically it's just this molecule that's produced, that actually gets converted into ethanol and carbon dioxide. So ethanol, which is alcohol, will be like a byproduct of the production. And carbon dioxide is also a byproduct. So the whole point of putting like the balloon over is to prevent oxygen.
Yeah.
So carbon dioxide collects in the balloon.
Okay, thank you.
Ruby.
Yeah, so maybe I missed the first part, but what area of the sustainability aspect the yeast experiment are trying to address again?
Group, anyone want to talk?
Yeah.
Yeah, where are the penguins?
Sorry about that. I mean, I was just trying to let her do a lot of the talking because she was really excited about it. So, okay. So in terms of the sustainability, we realized that, so yeast is so everything you'll know when you read our lab manual, but every single one of our materials are very easy to combine and they are very inexpensive usually. And most of all, there isn't very much waste that is created from this whole experiment. So what we realized was that in communities like the Philippines and things like that, we wanted to maximize the education that could be taught from an exercise like this while minimizing as much costs and pollution that could be created from something like this. So we realized that traditional classroom experience might not only require a lot of standardized, like specific equipment, scientific equipment. So I was taking a look at the curriculums, and they required a lot of chemicals. They required a lot of glass and glassware and all that, all of the such. But we realized that without using any of that, we can have really good, valuable lessons in terms of microbiology and other curriculums in the life science, other topics in the life science curriculum. So because our project utilizes mainly yeast and all of its reactions, and using very cheap, easily ready to combine materials such as plastic bottles, balloons, and things like that, we realized that our experiment, our lab manual can have lots of sustainability. It's very easy to come by everything. And also we want to talk about just teaching the children or teaching the students about the possibility of what this can be used as like an energy source in the future. We wanted students also to be like motivated in terms of like industry and things like that.
Okay, so in a way, it's quality education. So it's a number 4.
So we want to maximize the quality of education while minimizing the price of education.
Yes. Leo, right behind you.
Thank you very much. That was a good presentation. Thank you for doing that. We will be moving on to our next group. So please, for this group, you can just wait in about 15 minutes or so, actually maybe 10 minutes or so. We'll start our stream on Twitch, so you can hop over there to wait for the closing ceremony.
Okay, thank you.
All right, thank you very much for your work this weekend.
Thank you.
Okay. Judges, please enter your penultimate scores.
The next team isn't here yet either, so we are a little bit early.
We are running early. I hope they got whatever they need to get done.
think they were scheduled for 3:50, so I might have to wait another minute or two.
So
I will
thank you
also not
for
give-
entering the score. So, yeah, I'm going to just take this opportunity to thank all the judges for taking 3 hours out of their Sunday to join us. We hope you had a bit of fun.
Absolutely.
And so it's always heartwarming to see these students come in and with practically no idea, and then after a couple of weekends, put something out that is reasonable to be implemented. We'll need to tweak them, but I think a lot of them are seem fun to me.
They're great.
Yeah, it's for a wonderful purpose. I mean, I think it's great that they're using their science backgrounds and their energy to do this. I mean, it's a perfect delegation exercise.
Yes. So yeah, judges, you're more than welcome to stay and watch the prize ceremony, but we will be emailing you the results anyway. And for those of you who shared comments for groups, we will also be sharing those along with their comments about their manuals to the group so that they know what parts they scored well in and what parts they were more weak in. But yeah, well.
That's good. Is there going to be a possibility of seeing some of these in action, down the road over the next year or so?
I hope so.
That would be amazing to see some of it. Yeah.
Yeah, our idea was from the last year was that, we could have taken some videos or taken some pictures of the results from last year in the Philippines or in Guyana. That was our target country last year. And then use that to motivate people this year. But unfortunately, we weren't able to travel to Guyana this year.
Yeah.
But we'll see what we can do. And, we have some plans to see whether we can transmit some of this information and knowledge just online to.
Or Izzy, you can just come with us when we go to the Philippines and then you can see it in action.
I would love to. I'm putting that on my bucket list.
Yeah, we're always looking for additional people to help us do these things. And yeah.
Oh, that would be wonderful.
Unfortunately, I see Angie has dropped her connection, but I also see the guests coming in. So I'll admit them now. And hopefully Angie returns.
And she's the host. Yeah.
She's
back.
I was just going
her
to say,
Wi-Fi has crashed.
I was just going to say that it's been a lot of fun listening to young people's thinking around education initiatives, because this has been sort of my own professional career's life passion work, right? So to see young people really excited about doing this kind of work gets me very excited.
Agree.
I echo that.
And just to note that we had people because it is online. We had people participating all around the world. So not only across Canada, but we had people in Taiwan, China, Qatar, maybe a few other places. So it's funny to see some teams get to work 24 hours a day.
Really?
Yeah.
Oh, that's great.
So I don't know if Angie's fully back, but I will just ask if scientists for education, is your entire team here yet?
So it's just my one teammate and I. So again, because we're all over the world, due to time differences, it was kind of hard to get everyone on the call, but they're really excited for us to present it for you guys. So hopefully.
So I will start your timer now. So whenever you're ready, if you have any slides, you can put that on the screen right now.
Okay, so we did actually have slides, but we thought that since we're going to be focusing on the demo and just kind of getting our message across, we might as well just talk to you guys. So I'll start off with saying hello, everyone. We are the Scientists for Education team, and today we're presenting the Gene Machine, which addresses genetic engineering and just genes in general to the curriculum of the grade 10 to 11, as well as kind of the younger age groups in the Philippines. And we're looking specifically at genetic engineering focusing on insulin production, because we thought we could really tie that to the idea of diabetes, which is very prominent within the Philippines and has a lot of people have been diagnosed as diabetics within the Philippines. So it's definitely a problem that's very topical there. So it's really important to learn about the insulin production that the medical field relies on to then supply it to a lot of diabetic patients. And so to do so, the kind of main thing that we were looking at as a group was to get a lot of the heavy concepts within genetic engineering in a very easy kind of hands-on experience that makes it really fun, just because all of us struggled with the idea of genetic engineering. Just hearing that kind of makes you think, oh, it's such a complicated concept. So we really wanted to have this simple, fun idea that we could do that could get students on board and then give the teachers a lot of space to add as much content as they saw fit to a lot of different age groups and to kind of add on to that. And so with that, we looked at kind of three main lesson developments. The first one being the bacteria and the plasmid itself, and looking at genes there. And then we looked at genetic engineering and the students' ability to kind of use their scissors as restriction enzymes and cut up the plasmid and kind of add another piece to it, acting as the human insulin gene. And then their ability to enter that gene into a bacteria and produce insulin. So they see the output of kind of all the work that they've put in. And that part really relies on a team effort and kind of the group work of the class, which again represents this idea of a strengthened medical field and ties in with the SDG 3 of access to healthcare and kind of the interlinking of a lot of SDGs. And through that way, they could kind of work together, again, other than their individual project, but together to produce that insulin and see that you need a lot of it and you need a lot of people working on it and you need a lot of bacteria to produce it as well. So I guess what I'll kind of quickly go through is a bit of the aspects of our project. So this is actually the plasmid that we are using. So it's just a cardboard one. But what the students will be able to do is basically cut out this piece right here. So I've already cut it just for time's sake. And it has a side on the end that they'll be able to add, which has Velcro right over here. And so this will act as the target gene or the human insulin gene that they're adding in. So they add it onto the piece and that's your recombinant plasmid that they can then insert and then produce insulin. To do so and to increase the kind of team effort aspect, we want the students to be able to insert it into this right over here and then kind of pile them on so that they see, oh, it's all of us kind of working together and it's all our individual projects joining up. And then what I have, and I hope you guys can see this, I know there's a plant, but this is the best background I could do. What we have is basically the collection mechanism right over here. And right at the top, it's a foam piece. Let me quickly move you guys. So we have a foam piece right over here with a syringe attached that the teacher can then add kind of colored water into it just to make things fun, maybe decorate it from the top however they wish, or kind of things that really resonate with the kids in class. And then as a team effort, and they could fill this as many times as they want. And so it can easily be done again. And then they could basically just press the syringe down, and what we see is that it will start to collect water at the bottom, and they can start to basically just measure how much insulin they're making as like with time or as the group progresses. So just to kind of go over the general aspect, we really wanted to go through all the hard conceptual areas of genetic engineering and allow the teacher to explain the steps as the students work through, but make it kind of really simple to put together and to piece and to cut up, because you don't want like a project that discourages your students to approach such a difficult conceptual kind of area, but you definitely want them to be engaged and for it to be fun. And we also wanted to leave a lot of space for the teacher to kind of include a lot of the aspects that relate to the class itself or that they see fit to the students in that class. And so in that way, they kind of have a lot of control over the elements they can add to the box, or even getting their students to add a lot of the decorative elements of a bacteria and what makes a bacteria. So yeah, the kind of nutshell of our project is genetic engineering in a very simple way to put together from the teacher's perspective and the student's perspective, but with a lot of room for kind of conceptual input at every stage the student pulls through, leaving a lot of space for discussion as well. Thank you.
Thank you very much for that. Judges. Ruby?
Oh yeah, thank you, Shad. I really like the demonstration. I also struggled with genetic engineering, so I'm glad that you found a very intuitive way. But just explain to me again. I did not see the connection about the water collection. Like, like you have the the recombinant plasmid that you were talking about is clear, but then you kind of like snag it to the syringe, and then you talk about water being collected. Can you explain what it meant or the implications of that?
Yeah, sorry, I guess I kind of rushed through that part, but generally we wanted the water to act as the insulin that they're producing. So we wanted them to actually see the output of the experiment rather than just give them this idea of, oh yeah, you made the recombinant plasmid and that's how we're going to make like insulin, right? We wanted them to see that you're putting this plasmid into a bacteria and then that there's a process in between which kind of acts as your like foam that's happening there, right? And the teacher can then explain as well that this is the cell membrane and this is the cell wall, right? That encompass the foam. And then the output of that is, oh, look, through all of your hard work and all these plasmids, you're actually making the insulin at the bottom. And then you see that liquid that we're making, we can then use it for medication and we could treat diabetes and kind of get into that topic of this is what we're using to treat people.
Okay, thank you.
Do we have any other questions from the judges? All right, seeing none, we're done with our final pitches for the 2020 Hackathon for Science Education. Angie, are you here?
Yes, I am. My Wi-Fi cut out for a little bit, but we're back.
So thank you very much to your team for your hard work, again, through the day and night over the past 2 weeks. We will be beginning the closing ceremony in about a minute or two. So we're going to let you go. Angie will quickly switch over to the other feed. And everybody, if you want to stay watching, you can go to Twitch, which is twitch.tv/Pueblo_science. And I'm going to drop that link here onto the chat. But thank you very much for all of your time and for all your great questions and for all of your